Now let's discuss the viral envelopes. Do you remember the two important functions of the viral envelope? Well, one, it aids in host cell invasion of the virus, and two, it protects the virus from our immune system. So another question for you, are envelope viruses more sturdy than naked viruses? So you may think that since envelope viruses have an extra layer, they are more stable. But in fact, this is not true. Envelope viruses are more susceptible to damage and destruction in the presence of detergents, extreme pH, extreme temperature, and organic solvents such as ether. Naked viruses, on the other hand, are more stable in these environments because they do not have a lipid bilayer that is destroyed in these conditions. So in clinical application, when wiping down a surface with sterile wipes, which viruses are more easily removed, naked or enveloped? Well, you got it. It was the envelope viruses. So do you remember where the envelope viruses get their envelopes? Well, as you can see in this image, envelope viruses acquire their envelopes from the plasma membrane of the host cell. When the virus infects the cell, it releases its enzymes and genomic material, and it begins replicating its material. Once it produces its important proteins and is ready to exit the cell, the virus packages itself to its capsid protein, and then, when it's leaving, it takes part of the plasma membrane with it. The exception to this is with the herpes viruses, which actually gets their envelope from the inner nuclear membrane. So now how do we remember which viruses are enveloped and which are not? Well, we can always trust the creative authors of first aid to provide us with some handy-dandy mnemonics. In total, there are 8 naked viruses, 4 are DNA and 4 are RNA. The DNA viruses include the papilloma, adeno, parvo, and polyomavirus. Think of PAP and think of the pap smear. The 4 RNA viruses are calicivirus, picornavirus, reovirus, and hebivirus. Think of CPR-H. So, let's give pap smears and CPR to naked herpes.